of the year from this special interest group. It's hosted by the PSI, and thanks to Georgie for all the help with setting up the technical and logistical things about it. We have two widely known presenters today. The first presenter is George Quarty. He's also a member of both the European version and the North American version of the benefit risk. So in Europe, that's the PSI and the FSPI special interest group, and the QSPI counterpart in North America. George is working for Ross Genentech and has over 25 years of experience in statistics and research, and he's currently responsible for leading major innovation and enablement projects related to benefit risks. And he has also been included in projects like IMI Protect and other things. He's currently the co-director of the IMI EU2P program on benefit risk assessment of medicines. And he'll go first with his presentation about benefit risk assessment via case studies, key consideration, and best practices. As the second presenter we have today, Jason Wong. He'll talk about the emerging fields of benefit risk and health technology assessments, or shortly HTA. He's a principal statistician at Celgene, and like George, has over 25 years of experience as a statistician in many areas of pharmaceutical research. He has published more than 50 peer-reviewed papers and a book on exposure response modeling. He has also worked on health economics and outcomes research in academic institutes for several years, and that gives him a very, very good background to talk about this topic. He's leading now a working group on clinical trial exploration for extrapolation for HTA, and is the coordinator for the BEST-BY joint working group for benefit risk assessment and HTA. So kind of the joint group from Chrissy Fletcher's group and my group. So thanks a lot. So George, it's your floor.
Okay, thank you, Alexander, for the introduction. My topic for today is benefit risk assessment via case studies, key considerations, and best practices. This is actually a collaborative work between myself, Dr. Chi Jian from Amgen, and Willie He from Merck. And this actually forms part of a book chapter on benefit risk assessment of medicine that was published just summer of last year. So the objective of this presentation is, first of all, to discuss key considerations for benefit risk evaluation using two well-known case studies. And I'll start with the background of each case study and provide key benefit risk considerations for each, starting from the first case study, that is dabigatran, and the focus here will be on subgroup profiling. The second case study is about rivaroxaban or Xarelto, and the key consideration here is burden of missing data. And I'm going to finish off with lessons learned and best practices in benefit risk evaluation. So, starting with the first case study.
George, I think we cannot see your slides at the moment. I think it's still loading to come up.
Oh, I see. Is it? Can you see the slides?
Press show screen at the top of your toolbar.
Yes, I've done that. can you see the screen?
No.
You can. You can or you can't.
I can't.
Share. Okay. Can you see my screen?
Yes, now we can see it.
Oh, so I have to start again. Okay. So
can you press play? Can you press play?
Above the Roche sign. Perfect.
Great. Thanks a
okay.
lot.
Sorry about that. Okay, so benefit risk assessment via case studies, key consideration and best practices. As I said, this is a collaboration between myself and Dr. Jian from Amgen and Willie He from Merck. The objective here is to discuss key considerations for benefit risk evaluation using two well-known case studies. The first case study I'm going to talk about is about dabigatran, and the focus here is on subgroup benefit risk profiling. The second case study is about rivaroxaban, and the focus will be on burden of missing data. Then I'm going to finish off with lessons learned and best practices in benefit risk evaluation. So I'll kick off with the first case study, and this is a publication that came up in the New England Journal of Medicine. And it's about why the FDA approved a higher but not a lower dose of dabigatran. So to give you a little background of dabigatran, 110 milligram versus 150 milligram. This drug, dabigatran, is a blood thinner, and that was approved in October of 2010. And it's indicated to reduce the risk of stroke and systemic embolism in patients with atrial fibrillation. The benefit of this drug is prevention of strokes, heart attack, and death, and the risk is bleeding. So the evidence of efficacy and safety was established on the basis of a very large phase 3 active control study in patients with AF. About 18,000 patients with at least one additional risk factor for stroke. The study was randomized into 1:1:1 randomization of warfarin, dabigatran 110 milligram and dabigatran 150 milligram. The primary hypothesis for this study is whether dabigatran at either dose 110 and 150 was non-inferior to warfarin in preventing stroke and systemic embolism. So this shows the efficacy and major safety outcomes in this large study result. So as you can see, this data shows for all patients with at least one event, and the analyses are based on time to first event. As you can see from the top arrow and then the bottom arrow, the top arrow shows the primary analysis and endpoint, and then for efficacy, and then the bottom shows the major bleeding episode, which is the safety, primary safety endpoint. So 150 milligram dose reduced the risk of stroke and systemic embolism more than the 110 milligram, but did not cause more bleeding. So now looking at the overall benefit risk profile of this drug. So for each 1,000 patients treated with dabigatran instead of warfarin for 12 months, the 110 milligram twice a day dose will prevent 2 strokes, i.e. 4 fewer hemorrhagic and 2 excess ischemic stroke, and will also cause less than 1 systemic embolism. But it will cause 7 fewer non-fatal bleeds, 6 fewer life-threatening bleeds, 5 fewer intracranial bleeds, and hemorrhages. Compared to the 150 milligram, The 150 milligram will prevent 6 strokes, and also similarly less than one systemic embolism. But the risk here is different from the 110 milligram. We will have 3 fewer non-fatal bleeds, 4 fewer life-threatening bleeds, and 4 fewer intracranial bleeds. So in summary, if you treat 1,000 patients with dabigatran instead of warfarin, you have the 110 milligram will be non-inferior on stroke prevention, but superior on bleeding, compared to the 150 milligram, which will be superior on stroke prevention, but non-inferior on bleeding. So based on this result, both the 110 and the 150 milligram twice a day dose met the evidentiary standards for safety and effectiveness. But if the sponsor had studied either dose alone, according to the agency, they might approve both. But the question is whether to approve both or let practitioners choose between the 110 and 150 milligram dose. So the review team decided to identify within the large REALIZE study a patient population for whom the 110 milligram dabigatran dose suggests superiority to the 150 milligram. So they resorted to subgroup benefit risk profiling. So they picked elderly patients over 75 and older, and this population is about 40% of the entire REALIZE study. So what's the rationale for this? For choosing the elderly patients here? Atrial fibrillation is largely disease of the elderly, and therefore the risk of stroke and bleeding we expect to increase with age. So among these 40% of patients in the REALIZE who were 75 years of age and older, as you can see, the rate of stroke or systemic embolism was lower with the 150 milligram of dabigatran compared with the 110 milligram of dabigatran. But the rate of bleeding was higher. Note that if stroke or systemic embolism and major hemorrhage were considered equally undesirable, these rates will indicate similar benefit risk profile for the 2 doses. But however, stroke normally causes irreversible effects or damage to the myocardium, and therefore will be considered by clinicians to have a greater clinical significance than non-fatal bleeding. And therefore, any benefit risk assessment in which stroke and systemic embolism are given more weight than non-fatal bleeding should find the higher dose more favorable in the elderly patients. So the FDA also considered patients with impaired renal function, those patients with creatinine clearance between 30 and 50. So the rationale here is that dabigatran is cleared primarily by the kidneys, and therefore patients with impaired renal function could potentially benefit from a lower dose. So looking at the results, among patients with impaired renal function, the rate of stroke or systemic embolism with 150 milligram was approximately half that with the 110 milligram, and the rate of bleeding was no greater. And note that even in a population exposed to a relatively high concentrations of dabigatran, as in the elderly, the 150 milligram dose had a superior benefit risk balance. And finally, the FDA decided to look at patients with impaired renal, patients with previous bleeding, or patients who are at risk of bleeding. And as you can see, patients with bleeding episode during the study, so among those patients who were at higher risk of bleeding because of previous bleeding, the 110, there's no difference between the 110 milligram and 150 milligram as well as the warfarin. So none of the subgroup population shows the benefit risk profile more favorable for the lower dose, and the FDA approved only the higher dose of 150 milligram twice a day. And the FDA noted that if physicians, based on these subgroup analysis, the results of this subgroup analysis, if physicians are given the free ride to prescribe the less effective regimen, this may lead to unnecessary strokes and disability. So the next slide, I'm just going to summarize some of the key considerations made by the FDA in this benefit risk assessment. So this case study highlights a number of issues that need to be considered in benefit risk assessment. The first one I would like to talk about is endpoint selection. So when considering benefit risk assessment, careful consideration should be given to the endpoint chosen for the assessment. First of all, we should try to avoid something known as double counting. This is same event that contributes to multiple outcomes. For example, death counted in both PFS and OS, etc. And this may lead to correlation between endpoints. So double counting and correlation between endpoints may exaggerate or undermine some attributes of benefit risk profile and may potentially distort benefit risk profile. So how do we handle this? We can do that by using a single composite, or one efficacy and one safety endpoint, or we could even define a new global benefit risk score. The FDA resolved this issue by focusing primarily on endpoints that were fatal or cause irreversible harm to the patient. So they selected endpoints that are clinically important as the second point. In endpoint selection, we could use value tree or effects table to facilitate the selection of endpoints. And there are many challenges, as we know, in endpoint selection, because we will have various or disparate data sources, we have different metrics and scales across endpoints, etc., etc. Therefore, input from clinical and safety is key to endpoint selection. The second consideration here is weighting or relative importance of endpoints. As we know, different endpoints often carry different level of importance and weights. Therefore, assigning weight is, as we know, is value judgment and very, very subjective. So we could weight either qualitatively by ranking endpoints in a qualitative way or quantitatively. The important thing to note here is that the perspective of the stakeholder is key in this weighting. As you can see from the slide, from this particular example, from the regulator perspective, they are most concerned with stroke and bleeding events that are fatal or cause irreversible damage. But later research shows that substantial variation exists between patients' willingness to tolerate bleeding risk in exchange for stroke prevention. In terms of relative importance, type of weighting, there are various methods for doing that. And I've listed quite a few: point allocation, discrete choice experiments, using swing weighting, and as well as patient preference. The third consideration is the choice of a benefit-risk metric to use in the analysis. It's important to consider using a relative risk with absolute risk for a full benefit-risk evaluation and for communicating benefit-risk results. For example, a 40% relative risk increase in bleeding may generate a concern without considering the absolute risk and how rare these events may occur. And there are various metrics to use. For example, we can use risk difference, excess number of events, number needed to treat and harm, net clinical benefit, or we can resort to exposure-adjusted risk analysis. And finally, subgroup identification. So this is what the FDA did in approving the 150 milligram instead of the 110 milligram. So treatment effects in subgroups of patients defined by some characteristic. So there are various methods for doing this subgroup analysis. The FDA resorted to use a simple 2x2 table, but this can be done using various methods like recursive partitioning, regression-based approaches, or you can use MCDA type index to do the subgroup profiling. I'm going to start with the second case study, and this case study is about the ATLAS, also a very big study in patients with acute coronary syndrome and with a drug called rivaroxaban. So just a little bit background of the rivaroxaban. This drug is an oral anticoagulant, also a blood thinner just like dabigatran that directly and selectively inhibits factor XA. And this drug previously approved for 4 clinical indications in the US. The potential benefit for this drug is also to reduce the risk of recurrent arteriothrombotic events in patients with ACS, and the risk is bleeding. So the evidence of efficacy was established on the basis of a big ATLAS ACS2 TIMI study, which is a single pivotal phase 3 double-blind placebo-controlled study, very huge, over 15,500 patients, and the follow-up is over 31 months. And the rationale is to determine if rivaroxaban, when added to the antiplatelet therapy, is effective at reducing the risk of composite cardiovascular of death, MI, or stroke compared to placebo in these patients. So the primary endpoint is a composite of CV, death, MI, and stroke. Primary safety is first occurrence of this TIMI major bleeding that is not associated with coronary artery bypass graft surgery. The key here is the design of the study. So the primary evaluation strategy here is a so-called modified intention to treat analysis. And by that I mean randomized patients and the endpoints that occurred after randomization and no later than the completion of the treatment phase of the study, 30 days after early permanent discontinuation of the study of the drug, or 30 days after randomization for patients who did not receive the drug. So what's the primary efficacy results? As you can see, the treatment is very, very clear and very staggering. Treatment with rivaroxaban significantly reduced the primary efficacy endpoint of death from cardiovascular causes, MI, or stroke as compared with placebo in all the various strata and combination doses. The primary safety analysis is also very, very clear. Treatment with rivaroxaban resulted in higher rates of bleeding overall compared to with placebo in both the 2.5 milligrams and the 5 milligram twice a day dose. So what So I'm just going to give you a summary of a key benefit risk consideration. As you can see, despite this robust efficacy data, there were several key issues that were brought up that challenges the validity of this trial. The key is the study design flaw that led to a lot of missing data, basically. So I'll just go through some of the key considerations here. First, the issue with study design. The primary analysis was based on this modified ITT, i.e. events that occurred while subjects were taking study drug plus 30 days after early discontinuation. The FDA actually agreed to this a priori in the sense that the sponsor stated that all efforts will be expended in capturing the status of all subjects at the end of the study. The issue here is that the data needed for reliable ITT analysis are not available due to the incomplete follow-up that occurred. So this is a comment that came from one advisory committee panelist, and this summarized the major issue with this study design. And I read, the decision to use this modified ITT as the endpoint, I believe, had a profound impact here. And I think what happens when you say the primary endpoint is 30 days after you stop study drug is you are telling the investigators and you are even telling patients that you don't care so much about what happens later on. I think that's why they had such a large withdrawal of consent rate. I think it was preordained by the use of this so-called MITT, which is really an on-treatment analysis. And so I think it colored the trial in more ways we could never recover from, because we are never going to ever see an ITT data. So let's now go in more detail about what actually happened. Looking at the burden of missing data from what was presented, over 15.5% prematurely discontinued and 8.3% withdrew consent. Vital status was not ascertained in 86% of patients who withdrew consent. So this may lead to serious threat to validity of the study. Also, the number of patients with unknown vital status exceeded the total number of primary endpoint events. And this may also lead to a very questionable result. Thirdly, the difference in the missing data nearly matched the difference in the primary outcome. And this also leads to ample opportunity to amplify or obscure any true difference in the endpoints. There were also differential dropouts. Missing data are differential by treatment group. Results bias related to treatment efficacy or tolerability, and this may lead to informative censoring and might complicate interpretation of the results. So in the end, the sponsor company submitted supplemental NDAs and the applications were reviewed by the FDA Cardiovascular and Renal Drug Advisory Committee on May 23, 2012, and then again in January of 2014, respectively, and each time the company failed to gain recommendation for approval by the FDA. So what did we learn from this case study? First of all, it is important to recognize the study design and trial conduct impact on missing data during benefit risk assessment. Also, informative censoring was likely to have occurred due to the study design flaw that I just described. Missing data, as we all know, can complicate interpretation or even invalidate an otherwise important study. Although as statistician, we know imputation of missing data allows the analysis to conform to an ITT. It requires, or it may require, plausible assumptions like missing at random that may be difficult to verify. Thus, prevention of missingness rather than treatment remains the optimal approach to limit the problem and subsequently enhance the credibility of this study. Fourthly, safety signals often emerge with long-term follow-up, and it is crucial to collect long-term outcome information. In the Atlas TIMI 1551 trial was designed essentially as a trial on treatment analysis to address primary efficacy objective. And for this type of trial, ITT principles should always be followed as a general rule of engagement. So what are some of the best practices in benefit risk assessment? First of all, we need to analyze the condition in detail and treatment options in terms of unmet medical need. This can be informed by incorporating patient views or incorporating views from patients on their condition. Endpoint selection and outcome definition is also very, very key in benefit risk evaluation. Weighting is also very important, considering the clinical impact of the choosing endpoints. Uncertainty evaluation benefit risk assessment is all about uncertainty evaluation. And as you can see from this particular, from the rivaroxaban example, study design flaw and missing data actually cause the drug not to be approved by the FDA. The choice of metric as well as doing subgroup identification is key in any benefit risk evaluation, as well as incorporating patient preference information and also risk evaluation and mitigation strategy. All this will form part of any structured systematic benefit risk framework. And as you can see, there are quite a few around. There is FDA benefit risk framework. There is the benefit risk benefit risk assessment to BRAC framework. There's also EMA effects table, and there's also SAYS universal measures for benefit risk assessment framework. So that's the end of my presentation, and I'll hand over back to Alexander.
Thanks a lot. Thanks a lot, George. We'll take questions at the end, and I will directly hand over to Jason. Jason, can you share your screen? Very good. Yes, we can see your screen and the slide, what is regulatory benefit risk assessment?
Okay, I will go to, right. Okay, thank you very much, Alexander. Very glad to have the opportunity to talk about benefit risk and health technology assessment, these two emerging fields. And this is on behalf of the joint working group of BRA and HTA 6, and as a joint work with my colleagues in the joint working group, Susan Tolbert from Amgen and Sharu Mitisa from Merck. So I will give a very quick overview and also a few examples to show how BRA and HTA can work together, or how we can make them work together. So yeah, my disclaimer scan very quickly. For this one slide summary for what's BRA, I think I can skip that after George's talk, and just emphasize that BRA concentrate on clinical outcomes, not including economics and other considerations such as cost effectiveness, which is the key consideration in health technology assessment. So I move to what is health technology assessment, HTA. Well, by the name, it is a way of assessing health technologies. It covers medical, social, economic, and ASCO, sorry, some reason I jumped for another one, and ASCO ratios issues, and it provides the policymakers with objective information so that they can formulate health policies that are safe. Sorry, I can't see my screen for some reason. Sorry about that. And that does safe, effective, patient-focused, and cost-effective. And HTA should be transparent, unbiased, robust, and systematic. There are probably two distinct features in HTA. One is multiple issues to consider, multiple goals to achieve. Another is it should be transparent, unbiased, robust, and systematic. In fact, the BRA shares more or less the similar feature, so we can probably borrow our experience on BRA for better HTA. That includes using BRA to summarize the clinical side and also the methodology we use for BRA may be used for HTA as well. And so we organize this, all the multiple features, multiple goals into a kind of hierarchy structure. On the left-hand side is probably what we have already considered in the BRA as effectiveness, safety, and other things like health-related QOL. On the right-hand side are the factors we normally don't consider in BRA, that's economical and ASCO scopes that people should consider in HTA. So we have this type of multiple feature, multiple criteria to consider. So one powerful tool for that is the multi-criteria decision analysis, MCDA. We have been using that for BRA, so that's a tool to make decisions according to multiple conflicting criteria, multiple attributes. These are the terminology in MCDA for the outcomes, for example, the effectiveness, safety, and cost, and so on. So the key issue in MCDA is making a compromise, since with conflicting criteria you can't improve one without worsening the others, so the decision maker should make a proper compromise between criteria based on their relative importance. So how to make a proper compromise is kind of central piece in MCDA. And another layer of complexity is on the multiple decision makers in HTA, since they often have conflicting interests. So making a collective decision is another thing that I can't cover today. So just introduce one common but very simple method that is using multi-attribute value or utility functions to form an overall value, so the decision makers can compare this overall value to make decision, and the higher the better. So often we take the very simple approach, assuming that the attribute value can be added up together. And so this the I of A be the S attribute. For example, that's survival time under treatment A, and VI as the value function, and WI as weights, and the overall value would be the weighted sum over all the attributes we want to consider. So the weight represents the compromise, and the value converts the attribute into a sub-value that you can add together. Since, for example, the survival time can't be added up directly to, for example, the rate of toxicity. But there's an assumption behind that assume the weight and values for one attribute are not affected by the other attribute, which this is called preference independence, which might not actually hold for in some situations. So in the next few slides, I just gave some examples to show how the multi-attribute value functions work, and in some situations. The first one is the nice decision support unit report down to MCDA in HTA. This is a hypothetical example. We consider 5 attributes: cost effectiveness in terms of net benefit, equity, innovation, patient compliance, and quality of evidence. So the characteristics are the attributes for drug A and B. So how the value of, say, 16,000 come from for drug A. So assume I'm willing to pay 30K for a quality, and the drug A gave me 1 quality, but it only cost me 20K. Then I'm 10K is better off. In that sense, I have net benefit of 10K. That's how the value in money come from. And some of the attributes are straightforward equities in terms of percentage, and the innovations as categorical. And then you need the value function to convert that into the sub-values before adding them up. So 16K net benefit convert to 0.72, and 26 convert to 0.84. But the report is not clear with how this calculation was done. For example, the quality of evidence, these are the same category, but end up with different values. But anyway, that's the kind of shows how this works in kind of hypothetical situations. And we also have the cost, we also have the weight, and you can see the weight for cost effectiveness is far higher than all the others. But that's hypothetical, probably doesn't represent what NICE will really wait for your submission, for example. So having this value, then it is very straightforward to calculate the overall value. You just wait and add them up. So you have the value of drug A is slightly higher than drug B. Actually, if you only make decision based on cost effectiveness analysis, then actually drug B is better. I think the reason is the innovation which gives drug A about 1 unit advantage of drug B, so that makes the order changed. The second example is the ASCO value framework. That's the American Society of Clinical Oncology. They propose a conceptual model for solid tumors that incorporates clinical benefit, toxicity, symptom palliation, and others, and combine them into one score they call it net health benefit. There's a lengthy section on the scoring algorithm, but you can see that's actually very clear if you write it into the multi-attribute value formula. So you can see the value is 100 weight for efficacy value, 20 for safety, and there are a number of other items called bonus item you can claim. For example, long-term benefit is the you have 20 bonus points if you can show that the drug is increase the survival by 50% at the point of 2 times of median survival. So there's also a scheme to discount using the endpoint that is not as good as overall survival. For example, if you use progression-free survival, then you get 80 weight rather than 100 for efficacy, and then if you use overall response rates, then you might get 70, since there is a 30% discount on the weight. This is example for how to calculate the ASCO value. As use an example, enzalutamide for treating prostate cancer. The clinical benefit score is given by the hazard ratio for death. So 0.63 is 37% of risk reduction. That multiplied by 100, since that's overall survival, then you got 37. And toxicity score as the enzalutamide has 11% of toxicity rates that multiplied by 20, since that's toxicity, so that there's the negative sign, so it's 2.2 for toxicity bonus point. The tail curve is the long-term benefit, since that's on the progression-free survival, so that gets 16 rather than 20 because of the 20% discount. Palliation, they got 10, and health-related cure got another 10 bonus points, so that made up around 71 units of net health benefit. And they also listed the drug costs as 8,000 per month, but they didn't say how they balance the value and the cost. So one thought in here could be, can we use the net health benefit or the ASCO value as kind of a quality equivalent for cost benefit, cost effectiveness analysis? So that's probably something we can think about. And another example is quite different. That's the American College of Cardiology, American Heart Association. Instead of giving kind of framework with quantitative value, they propose a 3-level of value framework that's high, intermediate, and low. And they did give a reference to cost consideration. They set the high value may be set at say 50K, and the low value at 150K per quality added. So and also the value category supplemented by the level or the quality of evidence based on the scientific level of and the robustness of the data. I'm giving this example is because there are similar three-level value frameworks, like the HYPOTHETICAL use the three-level framework. The SEER also have a very similar three-level framework. And these slides give you how the classifications and works. So class 1 is the high value, class 2A is the intermediate, and 2B the low value, and level A, B, C are the quality of the evidence. But there is no specific guidance on how do you specify or classify your intervention into each of the value categories. So although there are some recommendations for the value of the evidence, for the level of the evidence. So okay, I think there are obviously many more activities that I can cover with in this 20 minutes time. So I just show kind of graphic illustration for the recent activities in BRA and HTA. So we can see that a lot of things going on. Many of them started about, say, around 5 years ago or something, and there are some new activities added. There are some we don't have a space in this slide, and so indeed the BRA, HCA are the emerging field. Yeah, the summary. So BRA HCA has been developing rapidly. There are still multiple challenges in methodology as well as application and implementation and collaboration between multiple lab functions, and as the key to success. And we pharma statisticians should play an important role in BRA and HTA. So, and finally, we have the joint working group for BRA and HTA. And let us know if you want to know more about the group. So yeah, again, that's the joint working group, joint work with Suzanne Charu and the member of the joint working group of BRA at HTA 6. Right, that's it. I hand over to Alexander, and happy to answer questions.
Thanks a lot to both presenters, George and Jason. I've already the first question from John McKellar. That's towards George. There are lots of places where different adjustments, models, and subgroups can be selected. How much justification for these choices is required? Can the sponsor influence the FDA there, and is there any need to do sensitivity analysis around all these different choices that you can have in terms of subgroups, models, and adjustments? George?
Yes, most definitely. Sensitivity analysis is key in this situation, I think. And in terms of choosing the subgroup, normally it's based on some predictive, some factors that are related to that particular drug or that particular area of study. So, so far as sponsor, a discrepancy between sponsor and the agency is concerned, and there's always going to be that issue there. So I agree that sensitivity analysis will be key in this situation.
And in terms of the pre-specification or different approaches in terms of choosing analysis, kind of logistic regression or alternative approaches to interpret the differences, are there any recommendations regarding this?
I mean, we will follow a normal, statistical subgroup analysis. What do? It's always good practice to pre-specify the analysis that you decide to do, basically. So I think the benefit risk assessment subgroup profiling is not different from that.
So what I found really interesting there is that I think it was a Dabika-Tran case where you had the risk differences between the drugs in terms of these different endpoints. Is that a common approach, or is it also very often used that relative measures like odds ratios and relative risks are used instead of this risk difference?
For benefit risk assessment, as I said, it's always important to use both. You can use the relative scale for the analysis, and you can use the absolute scales for the interpretation and presentation of benefit risk result.
So your experiences in terms of interpretation is how many more patients benefit or get harm is easier to communicate than relative
Yes.
risk? Okay.
Yes, absolutely.
Have you seen, so a couple of things that I've seen in the past, especially in terms of network meta-analysis, is that ranking is more and more used, so that from the Bayesian NMA outcome, the ranks and the surface under the cumulative ranks are used, so the SUCRA. Have you seen something like that to be used with regulators up to now, or is it something that is more used in the HTA space?
Is it me or Jason?
Well, I think that's probably a very good question for both.
Yeah. In terms of benefit risk, I mean, what I've seen in advisory committee, presentations are all qualitative type rating based on clinical significance or clinical importance. So most teams use qualitative scale and arrange the endpoints in order of clinical impact, basically. I haven't seen any quantitative methodology or quantitative scales used in this aspect. Jason?
Yeah, well, I think for HTA, since it's the tradition is to estimate the exact effect, that's well, people use the estimate quantitative ones to for the evaluation. That's probably different from BRA.
Thanks a lot. I haven't seen any further questions, and we are also at the top of the hour. So thanks a lot for both presenters for the presentation today. For all those who have attended, thanks for attending. If you found these informations useful, there will be another webinar next week. If you find it useful and you want to tell others about it, please do so. You can still register via the homepage of the Benefit Risk Special Interest Group, which is www.benefitriskassessment.com. Go there, find the webinar details like you did, and please pass it on to colleagues and friends. Thanks a lot, and have a nice day.
Thank you, Alexander. Thank you.
Thank you.
Thank you.